to sequence. So the nucleotide arrangement of a particular DNA fragment or gene of interest or the whole genome. The sequence of the base pair sequence of a given DNA fragment or the gene of interest or the genome. So the genome is the whole genetic makeup of an organ. Okay. So that determining or identifying the order of nucleotides present in the DNA sequence. Okay. So that in order to DNA sequence is the process of determining or identifying the order of nucleotides present in the DNA sequence. So that is the DNA sequencing. Okay, so the DNA fragments, the order of nucleotide present. And you know the nucleonitrogenous bases are the one we usually consider. So that when you consider this nucleotide, it consists of the three major components, the nitrogenous bases, there are 4 bases, and the sugar and the phosphate are the 3 ingredients of the DNA, so in which the adenine, thymine, cytosine, and guanine are bases. And again, the functional piece of DNA is known as gene, which encodes for the protein. Okay, when we call a gene, that will code for a protein. So that also we already know. So that for understanding the structure and function of a gene, it is very important to study the nucleotide sequence of it. So the DNA sequencing serves this purpose. Okay, so for understanding the structure and functions of a gene, we need to know the sequence of these different base pairs or the nucleotides. So to determine these nucleotide sequence of a given DNA fragment or the whole genome of an organism, we are going to use the DNA sequence. Okay, so the DNA sequencing will help us to determine the nucleotide arrangements in a given DNA fragment or the gene, or otherwise the whole genome. So that if you look at these milestones in the DNA sequencing process, in 1953, so that the discovery of the DNA double helix structure, and in the 1975, the first genome of a bacteriophage X174 was sequenced, and 1977, the chemical cleavage method to determine the DNA sequence was developed. So it was developed by the scientist Alan Maxson and Walter Gilbert. So this method is known as the Maxson-Gilbert method. And later they developed a chain termination method to determine the sequencing. So that was developed by the scientists Sanger and group. And in 1982, the gene bank was started. So the collection of different genetic materials, so the DNA sequences were stored. And next is the PCR, and you know the uses of the PCR. So it has several uses in the field of biotechnology, and that also a milestone as far as the biotechnology or the DNA sequencing is concerned. So that also happened in 1985, and the scientist responsible for this PCR technique is Kary Mullis. And when you come to 1987, the first automated DNA sequencing, so that automated sequencing, earlier they were sequenced, then manually we had to look at the base pair arrangements. So here everything is automated. Okay, so the first automated DNA sequencing was developed in 1987. The machine by automated sequencing machine by the ABI. And next we had the first capillary sequencing. So then the capillary sequencing, but within a single gel, you can read the different sequences, okay? So that the CAPILLAR thing also was developed somewhere in 1996. And 2003, the Human Genome Project was completed. And 2005, the first next generation sequencing method was developed. Yes, NGS means next generation sequence. So these are some sort of milestones as far as not biotechnology, but the DNA sequencing is concerned. Okay, as far as the DNA sequencing is concerned, some of the typical or the prominent changes that occurred at the different period is given here. The DNA sequencing. So it is a laboratory technique employed to know the correct DNA sequence by the sequential chemical reaction is known as the DNA sequence. So that you are going to determine the DNA sequence, so that these pair arrangements by the sequential chemical reaction. So that we are going to use a sequential chemical reactions. Okay, so one after the other is known as the DNA sequencing. And the laboratory processing and computational analysis both are the key processes in the DNA sequences. Okay, so you determine the DNA sequencing through some of the chemical reactions, and this data is going to be processed in the laboratory, and the computational analysis will give you the correct sequence of the different base pairs. So once the chemical reaction is completed, the machine generated data are sent to the computer lab. And in the process, the amplification done by each nucleotide is recorded. So the amplification can be in different forms. It can be sort of radio labeled emissions, or otherwise some of the fluorescence. So it means the color emissions. Okay. So each and every time one of them, the nucleotides is added to the growing DNA chain, then this signal will tell you what type of or which nucleotide was added at that particular time. So it can be automatically transferred in the form of signal, and these signals will be recorded in the computer, and later you'll be getting processed data, and through this computer analysis you can get the correct sequence. Okay, so in the process, the amplification done by each nucleotide is recorded in the form of signals which are collected by the machine and analyzed on the computer. This is the basic mechanism. The nucleotides are either radio labeled or fluorescence labeled. Okay, so the fluorescence means it will emit the colors. Radio labeled, they will emit the different length of the radio activity. Okay, but the radio labeled ones are not preferred nowadays because of the hazardness of the using of the radio labeled one, so that the radio labeled one, so with the time was replaced by the fluorescent labeled nucleotides. Okay. So now if you look at these steps involved in the DNA sequencing, so there are several steps involved in DNA sequencing. And so the general steps involved in the DNA sequencing are the sample preparation and PCR amplification of the target sequence, and amplification purification, and sequencing pre-preparation, and DNA sequencing and data analysis. So these are the major steps involved in the DNA sequence. Sample preparation, so that is the DNA extraction, and PCR amplification of the target sequence. Okay, so that we'll be getting minute quantity of this target sequence through the DNA extraction, so that you want to increase that one to a certain quantity, so that is needed for the analysis. And amplicants purification, so that after amplification in the PCR we have to again purify whether this DNA should be the DNA of interest should be purified. And then you go for the sequencing pre-preparation and the DNA sequencing and the data analysis. So that the first step in the DNA sequencing is the sample preparation. So here the DNA is the starting material. So we need to isolate the DNA first. Okay. So the DNA can be isolated either from the animal or the plant or bacteria or plasmid or environmental. DNA can be used for it. And quality of the DNA is the major concern for the sequencing. Okay, so that we have to get the good quality semen, good quality DNA for the sequencing. Okay, so the quality of the DNA is the major concern for the sequencing. So for that one, to get this good quality semen, the DNA can be extracted using this proteinase K or spin column DNA extraction methods are recommended. Okay, so the most commonly recommended method for the DNA extraction to get the good quality DNA is the proteinase K or spin column DNA extraction methods. So again, it will give the high yield as well. So that the high quantity and good quality DNA reduces the chance of reaction failure. Okay. So here the sequencing, this is not as we think. Okay. So that if the things are not in order and if there are some mistakes or something is of not good quality or the not adequate quantity of this particular DNA, etc., then the reaction will not occur. So that is known as the reaction failure. Okay, so that the high quantity and good quality DNA reduces the chances of the reaction failure. So that you have to have high quantity semen at the same time good quality DNA. High quality DNA and good quality DNA. So that will reduce the chance of reaction failure. So that how do you decide the quality of the semen? So you extract the DNA from the one of these methods mentioned above, and you decide the absorbent level of the sample under the radio wavelength of 260 nanometer and 280 nanometer. The absorbance level of the DNA samples will be determined. So that values between for the 260 nanometer to 280 nanometer should be approximately 1.8. Okay. So if this ratio is around 1.8, then the semen is of high quality. Okay. And at the same time, we should have adequate quantity of the DNA as well. So the minimum of 100 nanogram DNA is needed. That's the quantity. Okay. Yeah. Okay, so this is the one how do you determine the absorbance level of the different DNA samples. And you will subjecting these DNA samples at 260 nanometer. So where the absorbance level is the maximum. And again at the level of 280. So the level of absorbance at the 260 to 280 should give a value of 1.8. Then you are sure the DNA sample is a purified sample, okay, or good quality sample, okay. And again, the minimum quantity needed for the analysis, so the sequencing is minimum approximately 100 nanogram quantity is needed, okay. So that the first step is a sample preparation. So you extract the DNA and you decide the quality of the DNA and you get adequate quantity of DNA. It should be around 100 nanogram. Then you go for the PCR amplification of the target sequence. Okay, PCR amplification of the target sequence. Again, if you look at here, the amplification is the pivotal step in sequencing. So that we are going to amplify only our gene of interest, not all the genetic materials available. Okay. So only the gene of interest or the DNA sequence we wish to study is first amplified, and the rest of the DNA is discarded. But in case of the whole genome sequencing, the entire genome of an organism can be sequenced. Okay, so the whole genome, we need all the genome, but here we will target only our gene of interest, or otherwise or the DNA fragment we are interested in. Okay, only that portion will be amplified using this PCR. Okay. So that for which you have the primers, the forward primers and the reverse primers, so that we should know after the gene of interest in the DNA strand, what is a few bases in the starting site and the ending site. Okay, the forward primer site and the reverse primer site, and we will be making the primers complement to the base pairs of that particular DNA sequence. Then they will go and fix there. So after the primer fixing only, the chain extension program will occur. Okay, so that the other places the extension process will not occur. So the primers they go and join with our gene of interest. The starting part in part will facilitate only the PCR amplification of our target sequence. So there is a set of flanking primers anneal at the outer regions of the DNA sequence of interest. So therefore, unwanted DNA can't amplify. Okay, so the primers we should know in advance. So the sequence we should know in advance, not only the DNA of interest, but the DNA sequence before the DNA of interest, or the sequence of interest, and the DNA sequence after the sequence of interest. Accordingly, we will be taking the forward primers and the reverse primers. Okay. So a set of flanking primers anneal at the outer region of the DNA sequence of interest. So as a result, unwanted DNA can't amplify. Okay, so the primary annealing is going to occur. Using the standard PCR conditions, the amplification process is done. So usually we go for around 35 cycles and denature the DNA strand, double strand at 94 degrees Celsius. And the annealing process will occur at the temperature of 55 degrees Celsius to 65 degrees Celsius, and the extension 72 degrees Celsius. So these are the conditions for the PCR that you know. So that after the amplification, the PCR product is run on the 2% agarose gel along with the DNA ladder. And once the amplification is achieved, DNA purification is performed. Okay, so that you know the band where you are looking for your DNA fragment. Okay, so based on the ladder, you can locate your DNA of interest. Then that particular portion of the gel can be cut out, and the DNA can be purified. Okay. So after the amplification, the PCR product is run on a 2% agarose gel along with the DNA ladder. So when the amplification is achieved, DNA purification is going to be performed. So now the DNA purification, we did that one at the 260 and 290 nanometer wavelength. And we determine the absorbance of this sample. And when it comes to around 1.8, we said, okay, the DNA is of purified, good quality DNA. So now we had to go for the amplicant purification. So the amplicant purification means now the DNA of interest has been amplified using the PCR. So now you've got the amplified product. So that is known as the amplicant purification. So then why do we go for this purification? Because we already decided it is a good quality DNA by getting the ratio of 1.8. Then why should we go for the amplification purification? And if any contaminants are present in the DNA, amplification will not happen. So that then why we go for the amplification purification, because there could be some unbound primers and the primer dimers, unused Cat DNA polymerase, unused DNA templates. So these are the things needed for the PCR reaction, okay? And other unused PCR reaction buffer components can abort DNA sequencing. So that's why we go for the amplicant purification, okay? So that for the amplicant purification, the alcohol purification can't work. So we had to purify our PCR product with an spin column PCR amplicant purification kit to achieve the maximum purification. So that you have a kit nowadays, so that will give you the separated our gene of interest to the DNA fragment, a purified thing. So here the alcohol purification method also there, it won't work. What we have to purify our PCR product with the spin column PCR amplicant purification kit to achieve the maximum purification. So that after removing the debris, the PCR amplicant sent to the sequencing lab. Okay, so that now we go for the other step, the amplicant purification. Why do we go? So the reason to remove all these things from the system, and we want only our DNA fragment of interest. Okay. So next step is the sequencing pre-preparation. So that again here the sample pre-preparation is very crucial step during the DNA sequencing. So what do we do during this pre-preparation? And during this step, adapted DNA sequences are ligated to both DNA ends. Okay, so that we are going to add or ligate adapted DNA sequences to the both ends of our DNA of interest. So to the adapter, the primer anneals for doing amplification. So that you are going to add a node sequence of DNA, both sides of our DNA of interest or the DNA of the fragment. So that the adapted DNA sequence we know well in advance. So that we will be making that primers according to the complement to the adapter region. Okay, we will be making the primers complementary to the adapter region. Okay, so to the adapter, the primer anneals for doing amplification. So the amplification process in sequencing is similar to PCR. However, the nucleotides are either radio labeled or the fluorescent labeled. Okay, so here the nucleotides which are going to be added at the extension process is either radio labeled or fluorescent labeled. Okay, so every time it is added, a radio active things, or otherwise the color will be emitted. So with it, high fidelity DNA polymerase and other key ingredients are added in the reaction. And next is the DNA sequence. Okay. So this is the main process. We are going to look at what are the different methods of DNA sequencing are available. So the DNA sequencing, the prepared reaction tubes are placed into the sequencer machine. Okay, so now you have the sequencer machine. So you add all these things together here. So that you have the amplified things, the adapters added. And with that, you add these the primers and the DNA polymerase and the other key ingredients which are needed for the chain extension process will be added. So that is similar to the PCR, but here the nucleotides are either radio labeled or fluorescent labeled. Okay. So next is the DNA sequencing. So the prepared reaction tubes are placed into the sequencer machine. So that during the reaction in the sequencer, denaturation, annealing, extension occurs simultaneously. Okay, so these all are going to occur simultaneously in the DNA sequencer machine. Here, as we are using the labeled nucleotides, the signals produced during the reaction is recorded. Okay, so again, I told you the nucleotides every time A, T, C, G are colored, fluorescent labeled, different colors, or radio labeled. So here we are using the labeled nucleotides. So the signal produced during the reaction is recorded. So the signals of the addition of each complementary nucleotides are recorded by the machine, and the data is sent to the computer. Okay, so that every time the nucleotide is added, the radio labeled action. Otherwise, the colors will be emitted. So that will be recorded by the machine, and that will be sent to the computer. So the computer with the specific packages, we will analyze the data. So that analysis in the computer, the sequencing data is known as the bioinformatics. Okay. So that next is the data analysis. Once the whole DNA is sequenced, the result is saved into one unique file format. The inbuilt software processes the data and compare it with the available data. Okay, so that you might, for example, if you are comparing our DNA of interest with the sequencing our DNA of interest with the previously non-interest, and there could be some changes, okay, some deletions, insertions, sort of thing. So if there is any modification, you can identify. And similarly for the genome also, you have to have a reference genome, and you can see if there is any modifications in the genome as well. Okay. So the inbuilt software processes the data and compare it with the available data. So the sequence data is compared with the other available data by the software to find out the variations and other mutations present in a gene. Okay, so by comparing this one, you can find out if there is any variation in the DNA sequence we are interested in, or the genome we are looking at. Or otherwise, if there is any other mutation present in a gene, okay, then we can locate it. So if you look at here, these are the brief overview of an DNA sequencing. Okay. Okay. Sidambara Sakkarath pay maatha manika illadi. Okay, so when you look at these methods of DNA sequencing, I told you already there were some milestones in the DNA sequencing. So at different periods, different methods were developed for the DNA sequencing. So the very first method was Maxam-Gilbert method. So it was developed in 1977. And this method is also known as the chemical cleavage method. Okay, so that the names can appear in different forms if the question is there. So don't confuse, then don't say this, didn't give that one. Okay, so the Maxam-Gilbert method. So it was developed in 1977. And it is also known as the chemical cleavage method. Okay, so that the chemicals are going to cleave the DNA strand. And if you look at the brief principle of this Maxam-Gilbert method, so that the single-stranded DNA is cleaved at the specific locations with the help of the chemicals at a specific base location. And the fragments of DNA is then run on a polyacrylamide gel. Okay, polyacrylamide gel. Okay. So now the DNA is going to be fragmented with the different chemicals. These chemicals will fragment this DNA at the specific base location. For example, is CTG. Okay, some might cut at the A position. Some chemicals might cut the DNA at T position, and some might C and some might G. Okay, so these are that at the specific base locations. the different chemicals will cleave the DNA. As a result, the DNA is going to be fragmented. And the fragmented DNA is going to be run on a polyacrylamide gel. Then you will be getting a band pattern. So according to the label you used, the radio labeled or fluorescent label, you will be getting the bands, and based on that one, we will be deciding the sequence of our DNA fragment of interest. So that is the Maxwell Gilbert method. Okay. So now you see already 2 persons they left out. Okay. Later they will come. I am biotechnology wrong. Chennai miss didn't give the marks. Okay. Okay. So the single stranded DNA is cleaved at a specific location with the help of the chemicals at a specific base location. Polyacrylamide gel. That is the principle. Okay. So that as usual in the DNA sequence, so for the Maxwell Gilbert method as well, we have the steps. So the first step is the DNA extraction. And second, so the sequencing is going to be done only for the single stranded DNA. So the DNA is denatured using the heat denaturation method. And single stranded DNA is generated. So through the heat denaturing process, the single stranded DNA is going to be generated. And so the enzyme named phosphatase removes the phosphate from the DNA. So that you see the phosphate group is one of the component of our DNA strand. And so that what will happen, the enzyme phosphatase, when you add, that will remove the phosphate from the DNA. And you'll be adding another enzyme simultaneously known as the kinase. So that will add the radio labeled phosphorus in place of the normal phosphate. 32 labeled phosphate in place of the normal phosphate. So that this is how we are radio labeling, and not all the phosphate, but only the 5 carbon end of the DNA will be radio labeled. It is 32P phosphorus. So that you need for that one 2 enzymes, phosphatase and the kinase. Okay, so the DNA strand, when treated with the enzyme phosphatase and the kinase, and the normal phosphate at the 5 carbon end of the DNA will be removed. And at that place, the 32 phosphate is going to be replaced or put in place of the normal phosphate with the enzyme known as the kinase. Okay, so the radio labeling the DNA strand. So here, the Maxwell Gilbert method, they used the radio labeled method. So that's why in the latter part, this methods were not preferred because the radio labeling process. Okay, but anyway, here the enzyme named phosphatase removes the phosphate from the DNA, and simultaneously the kinase add the 32P to the 5 carbon end of it. So that again, the next step. So you have extracted the DNA and you have radio labeled denatured the DNA, and you radio labeled the DNA with 32P phosphate. And next, I told you the basic principle here. The different bases are going to be cleaved by 4 different chemicals. So that's why we call it the Maxwell Gilbert method, or otherwise chemical cleavage method. Okay, Maxwell Gilbert method, or otherwise we call it the chemical cleavage method. So the 4 different chemicals are used to cleave DNA at 4 different positions. So again, when I look at this literature, so they have been using different chemicals to the cleavage. So here one protocol is given anyway. So that hydrazine and the hydrogen sodium chloride are selectively attack the pyrimidine nucleotides, and while the dimethyl sulfate and piperidine attack the purine nucleotides. Okay, purine and pyrimidine nucleotides will be attacked by the hydrazine and hydrogen sodium chloride. And while the dimethyl sulfate and piperidine attack the purine nucleotides. Okay, so hydrogen P plus C and hydrogen sodium chloride only the C, and dimethyl sulfate A plus P and piperidine only the guanine. Okay. So what we do here in the Madsen-Gilbert method, an equal volume of 4 different single-stranded DNA samples taken into 4 different tubes. Okay. So you are going to take 4 different tubes, and into which equal amount of volume of single-stranded DNA is going to be added. Later, 4 different chemicals are going to be added to one chemical to one tube. Okay, so that now 4 tubes A, B, C, D, equal volume of the single-stranded DNA. And for the 4 tubes, 4 different chemicals are going to be added. So they will act on different base locations, and they will keep the DNA. Then the samples are incubated for some times and electrophoresed, so that you are going to run the polyacrylamide gel. Okay. And the results of the chemicals cleavage of 4 different tubes are shown in the figure below. So after that, what will happen? I'll show you the figure in just a second. Okay. So that autobiography is used to visualize the separation of the DNA fragments. So due to the radio-labeled 32P end of the DNA bands visualized through the autoradiography. So the autoradiography is that you are going to use the X-ray films. So the radio emissions will be passed to the X-ray film. Then you can see that bands. Okay, so that is the autoradiography. So now you could see our gene of interest is this one. Okay, our gene of interest is this. So this the equal quantity of this single-stranded DNA is added all 4 tubes. Okay, all 4 tubes. And so here the 4 different chemicals will cleave this particular DNA fragment at 4 different places. Okay, so here the cleavage at A. Okay, the cleavage at A. Okay. And the first component you can have here the ACCT. Okay, the cut is here. Okay. Then the second A here, so up to this level. So when you have this cut at the A, so you will be having these components, okay, these fragments, okay, these fragments. And now here the cut is at G, okay. So where are the G's? We'll be getting ACC, TAT, the first cut. The second G is here, then you will be having ACCT, AT, and GCA. Okay, the second G is here. And the third G is here, then you will be getting this much of extension. Okay, so when the cut is at G, you will be getting this. And when the cut is at, because even though it says A and G, they mention only A because this is the cut you are going to get the similar, despite of the tubes. If the cut is at G, these are the fragments you will be getting. Okay, so that A cut you will be getting these fragments, and this what is that? The G cut you will be getting these fragments. So when the cut is at C, so the first C, then A only, and similarly the AC, and these are the fragments you will be getting when the cut is at C. And here, even though the C is C and T, so if the cut is at the C place, these are the fragments we are going to get. So here, this is only for the cut is at T. Okay, so now, can you make a record of this strand? Okay, the target strand, and try on your own when the cut is at A place and when the cut is at G, when the cut is at C, and then when the cut is at T. I'll give you a few minutes, try to find out the fragments. Getting nothing. So that's why this fragment is not coming. So that when the cut is at A, so these are the fragments, so you'll be getting. When the cut is at G, these are the fragments you'll be getting. When the cut is at C, these are the fragments you'll be getting. And when the cut is at T, these are the fragments you'll be getting. Okay. So that now this is A tube. Okay. And this is G tube. Sorry. And this is for what? C, and this is for T. Okay. Then when you run the gel, these are the bands you will be getting. Okay, these are the bands you will be getting. And the reading is, okay, the reading is, how do I say? The smallest to the biggest one. Okay, the smallest to the biggest one. So where do you find this smallest fragment in the gel? Where do you find the smallest fragment in the gel? So this is the negative side, this is the positive side. So where do you find the smallest one? Near to the positive side or
negative side? Negative side. Negative side.
So you'll be finding that the smallest one towards the negative side. The electrophoresis principle is what? The electrophoresis principle is what? Can you recall it? So DNA is negatively charged or positively charged?
Negatively charged.
So that will move towards the positive side or negative side?
Positive side.
So that it will move towards the positive side. The smallest one will move fast. Yes or no?
Yes, ma'am.
Okay, so the smallest one will move fast so that it will be near to the positive side. So then you have to read it from the smallest to the biggest one. Okay, so this is the smallest one, that is A. And the next is, what is this? C. Okay. And here you see, you have two bands. Okay. So that the C, the third one is for the C and the fourth one is for the C plus T. Okay. So you have, and when you have the lines, the parallel lines at two places, you have to be careful. Okay. So this is an example. So the first one is A, there's no problem. And next is C, no problem. Third band, you have two parallel bands. So this cell is for the C and this is for C plus T. Okay, so in both places you are getting the band. But you see this is for C plus T, but you see in the C, there's a band, so that you will be having only one fragment at a time. Okay, so that this is C, it's not T. Okay, so even though you have the C plus T well, since you are having the C in the C alone well, so that this is C. Okay, so this is how you read it. Understood now? So when you have the bands for the C and C plus T, because you will be having only one band at a time or the one cut at a time, you have to decide whether it is C or T. So since you get the C band, so it is obvious the C is the cut was at C. So that since you are having the chemical to cleave at C and T as well, so this is not for T, this band represents C. So this is how you read when you have the parallel bands. Understood or not?
Yes, Miss.
Understood or not? Are all the members?
Yes, ma'am.
Okay, so this is how you read it. So that again you'll be going to the smallest to the biggest one. Okay, smallest to the biggest one. Then that will your what is that sequence? Okay, and again this one is from 5 prime end to the direction. The smallest is the 5 prime end, and it goes up the 3 prime end. Okay, so that is the sequence for the 5 prime end to the 3 prime end. So again we have on the signal strand here. So that now since you know the sequence, the complementary strand can be easily synthesized. So that you are getting the double strand sequence. Clear? Clear?
Yes, ma'am.
Okay, so that this is how you get that one. And again, if you look at the prospects of this Maxam-Gilbert method, suitable for DNA footprinting and DNA structural studies. Okay, and again here the purified DNA is directly used for the sequencing as far as this Maxam-Gilbert method is concerned. Okay, and as far as the constraints are concerned, so the scalability is poor. Okay, so that what is the length of the DNA fragment we can sequence is only 400 base pairs. Okay, so the scalability is poor. And again, it's a time consuming method. So you have to have 4 tubes, you have to have 4 chemicals, you have to run the gel, and you have to manually read the sequence. Okay, so that we have to do the sequencing manually. So that it is a time consuming method. So the most important constraint is it's less popular because of the use of the harmful radio labeled chemicals. Okay, so the 5 carbon end is going to be radio labeled, so that is human hazardous. So that is a major constraint of the Maxam-Gilbert method. Okay, otherwise there are three constraints. Scalability is poor, or only 400 base pairs can be sequenced, and it is a time consuming method. At the same time, it is less popular because of the use of the harmful radio labeled chemicals. Okay, so just to give you a feel, can you see the sound? Can you see
Yes.
and can you say
Yes, Miss.
huh?
Yes, miss.
Okay. Okay.
Okay, please listen carefully.
Yes, Miss. Audible. different chemicals for the cleavage. Okay, it is differing from the one I gave you. prospects or the advantages and constraints of the Maxam-Gilbert method. So the second milestone in the DNA sequencing is the Sanger sequence. Okay, so this is the, how do I say, this is the foundation. This is the foundation or the pillar for the rest of the DNA sequencing methods development. Okay, so this is the Sanger sequencing method. And here, if you look at this Sanger, so it's not a single scientist, but co-workers. Okay, so the Sanger and co-workers developed a chain termination method of DNA sequence. Okay, so there we looked at the Maxam-Gilbert or chemical cleavage method, but here the Sanger and co-workers developed a chain termination method of DNA sequencing, so that it can be called as the Sanger sequencing method, or otherwise it also can be called as the chain termination method, or otherwise it is also sugar, but in case of DNA, you have the deoxyribose. Here, in the carbon, this particular second carbon, the O group, O is removed, so that we call it a deoxyribose. Here, the O is present. Here, the O is removed, so that we call it deoxyribose. Okay, but in case of this dideoxyribose, what happens? The third carbon O also going to be removed. Okay. But what is the importance of this third carbon hydroxyl group is, okay, OH group is, this is needed for the development or establishment of a diester phosphate bond when a new nucleotide is added to the extending or growing chain of the DNA. This OH group is essential to create this diphosphate bond between this existing nucleotide and the forthcoming or incoming nucleotide. Okay, but in the dideoxynucleotide, what will happen? The O group is not there. The diphosphate bond cannot be formed. Okay, so that you see, usually the nucleotide with the deoxyribose will go and join with the existing or the growing chain of the DNA. Then the phosphodiester bond will be formed with the existing nucleotide and the incoming nucleotide. Okay, then the chain will keep on growing. But here what will happen is when the nucleotide come and added to the existing thing, there's no any other OH group to form the phosphodiester bond, so that the chain growing is going to be terminated. The chain will not go further. So that is known as the chain termination method. Okay, so the OH group which is needed for the further diester bond formation is not facilitated when a dideoxyribose nucleotide come and add to the chain. So this is the principle which is going to be used in the chain termination method, or the Sanger method, or the first generation method, or dideoxynucleotide method of the DNA sequencing. Okay, so that if you look at these dideoxynucleotides are different from the normal deoxynucleotides. It possesses the hydrogen group instead of the hydroxyl group in the DNTP. Okay, so only the hydrogen group instead of the hydroxyl group. But this hydroxyl group is important for the formation of the phosphodiester bond to keep on the chain is growing. Okay, so while the chain is growing, in place of the DT and B, if DD and TP come and join, the chain elongation will be stopped. So that is known as the chain termination method. So the chain is going to be terminated. There is no further elongation. Understood or not? Clear or not? Okay, for that one also, I will put you a video, then you can understand. So that the thing is that you have a growing chain. For example, assume, okay, the growing chain now you have the C. So next, the nucleotide that is going to add something, maybe T or something. So that then between the C and T, the phosphodiester bond will be formed. And then you will be having another OH group in the T side. So that will commit another new nucleotide and come and join with this one. But in case of this dideoxyribose, what happened? The O group is not there to form the phosphodiester bond, so that the further chain elongation is stopped. So that we call this method as a chain termination method. Okay. And again, this one will not happen every time. Once in a while, the DNA polymerase will add randomly. Okay. Randomly, it will add this DNTP instead of DNTP. So what instead of DNTP, when the DNTP is added, the chain growth is going to be stopped, or otherwise the chain is going to be terminated. So that is known as the chain termination method. So that principle is going to be used in the Sanger method. Okay. So that the phosphodiester bond can't form between two adjacent nucleotides due to the absence of the hydroxyl groups in the DDNTPs. So the nucleotide chain can't synthesize further, and here it is known as the chain termination method. Okay, so that is the principle used in the Sanger method. Okay. So again, if you look at as for the earlier steps involved in the sequencing, here also the first step is the DNA extraction. So using any of the DNA extraction protocols. For the DNA extraction, we have different protocols. Okay, then we can use one of those protocols for the DNA extraction. And the PCR amplification. So the PCR amplification, you know that you need the DNTPs, that is ACT, and here the additional you have the DDNTPs and the TAF DNA polymerase and the PCR buffer and the flanking primers. Okay, the forward and reverse primers. Okay, so the PCR amplification, these are the usually we need the primers, DNTPs, that is the deoxynucleotide, so nucleotide triphosphate. And but here in case of Sanger, we have additional component, that is the DDNTP, that is the dideoxynucleotide. So when in place of the DNTP, when this DDNTP goes in and added, the chain is going to be terminated. So that is the principle as far as Sanger method is concerned. So that then you will add all these things, then you will facilitate this PCR process. Then due to the addition of the DNTPs at random places of the DNA fragment of interest, we are going to get a different dense of the DNA fragments. Okay. So the identification of the amplification fragments, so that you can go for the using autoradiography and polyacrylamyl gel electrophoresis or the capillary gel electrophoresis. So this is sort of advanced method of this Sanger or modified method of the Sanger sequence. So that we start with the DNA extraction and the purification, so that a DNA extraction can be achieved using the proteinase K method or phenylchloroform DNA extraction method, or we can use ready to use silica column based kit method too. Okay, so that they have different methods for the DNA extraction. And the main aim of the DNA extraction method is to achieve the purity of the 1.8. So I already explained what is this 1.8. And the minimum quantity we need around 100 nanograms. So the next step is the PCR amplification is performed by deciding 4 different reactions. Okay, so these are the 4 different PCR reactions as we did earlier. But here the only difference is we are adding first tube instead of the chemical 1, we are adding DDATP, and the chemical 2 instead of with all the other PCR conditions for the tube 2, you are adding DDTP, that is a guanine DD and thymine DD and the cytosine DD. Okay, so this is not in a high quantity, but a small quantity of these DDCs will add to 4 different tubes. Understood? Clear? Yes or no? So this is the TAC DNA polymerase for the chain elongation. So these are the four deoxyribose nucleotides: adenine, guanine, cytosine, and thymine. Okay, and the PCR buffers and the primers. So these are the things needed for the PCR reaction. So that in addition to that one, we are adding a small quantity of the DDNTP as well. So the A tube, you are adding the DDATP, and the G tube, you are adding DDGTP, and for the T tube, you are adding DDTTP, and for the cytosine group, you are adding labeled. Okay, so these all are labeled. Okay, it labeled DD and labeled DDGTP and labeled DDTTP and labeled DDCTP. This is clear?
Yes, Miss.
Yes, Miss.
Other members?
Yes, Miss.
Okay.
Yes, Miss.
So each tube contains the same amount of the PCR reagents, but in each tube, extra DD and TPs are added as shown in the table. So the flanking primers are such a primer that binds to the. So we know we already decided the primers based on the adapters we have added to the DNA interest or the DNA sequence of interest. So the flanking primers are such a primer that binds to the regions near to our sequence of interest or the sequence what we are going to read. And next step, the Taq DNA polymerase at the DNTPs on the DNA strand. So the high fidelity DNA polymerase is not needed. Normal Taq polymerase expands the decoding DNA strand by addition of the DNTPs. So once it adds the DDNTP instead of DNTP, the chain expansion is stopped or terminated. So this is the major step as far as the Sanger sequences is concerned. So okay, instead of DNTP, every time it does not add is continuously, okay, randomly it will add, okay. So instead of DDNTP, when it pick this DDNTP, the chain expansion is going to be stopped or the chain is going to be terminated. So the termination process is completed in 4 different tubes for 4 different DDNTPs. So that in the, for example, in the DDATP tube, it terminates the chain at all the position where the DATPs are going to be bind. Okay, so that the probabilities the DDNTP can go and join. So wherever the DATPs can go and join, so the DDATP can go and join randomly because we have a number of DNA fragments. We amplified that one. Okay, so that the chances at every A places the DDATP will go and join randomly. So they will be getting different fragments of that particular DNA fragment. So the amplified PCR products are loaded onto the polyacrylamide gel electrophoresis. So the DNA fragments migrate into the gel based on the size of the fragment. So that is the basis or the basic for the gel electrophoresis. So the smaller fragments run faster towards the positive charge than the larger fragments. So that the PEG means polyacrylamide gel electrophoresis. Okay, polyacrylamide gel electrophoresis. Okay. So before the PEG run, the amplified DNA fragments are further denatured by heat. And so that depending upon the types of the labeling, the gel is then analyzed under UV light or the X-ray film. Okay, so that if it is the fluorescent, then the color or the radio label, then we have to look at the X-ray film. Okay. So this is similar to the earlier one where we had different chemicals. Okay, but here this is for the different DDNTPs. Okay, different DDNTPs, dideoxy nucleotides. Okay, so here too, can we make a note of this fragment? Thymine, and this should be cytosine, and this should be guanine, and this should be ATP. So, okay, for this tube, you are adding TTP, and here you are adding CTP, here you are adding GTP, and here you are adding ATP. So now you see when the ATP, not ATP, TTP is there, thymine, okay? So it will go and add the complementary base pair of A. Agree or not? Agree or not?
Yes, miss.
Okay, so when the first position, when the DDTP will come and add here, join with the chain is going to be terminated. So the chain, the only one A is in the chain. Understood? So the second possibility, the thymine come and join is this A. Okay, so here the chain is going to be terminated. So that the fact you have ACCT, it's not the card termination. So that the A also will come in, okay? And the third position, the thymine can come and join is here, this A. So the rest of the components are going to be in the fragment. So these are the different fragments you can get within the tube when you add this DDTP. Clear or not?
Yes,
Clear or
miss.
not?
Okay.
Huh? Okay, so similarly, the rest of the things also going to happen. Okay, so then so that you are getting the different fragments, then you run the gel electrophoresis. So similar to the previous one, you will be reading the what? Here you will be not having the parallel ones. Okay, so it will the smallest to largest. If you read, that is the sequence. Clear? Clear?
Yes, miss.
Yes, miss.
So this is sometimes this is known as the Sanger classical method, okay, the old method, classical, okay. So that when you look at this Sanger classical method, and again I told you this is the foundation or pillar for the many of the other DNA sequencing methods developed, okay. So that there were some advancements as far as the classical method of the Sanger sequencing is concerned. So the Sanger sequencing is the gold standard method, okay? It is referred to as the gold standard method for the research as well as in the diagnosis nowadays. Okay, so it still has been used as the gold standard method for the DNA sequencing. And because of its easy steps and high reproducibility, so you can multiply that one. Its automation has done easily, so that advancement means that now you can reproduce it easily and you can automate that one. So earlier you were doing the manual sequencing, but here the advancement you go for the automation. Okay, everything will be automated. And traditionally the results were interpreted on page manually, but now the scenario is changed. So that now the process is fully or partially automated. Okay, so the advancement in the Sanger method, classical method, is you have the partially automated method or the fully automated method. Okay, so that what happens in the advanced methods, a detector detects the fluorescent signal each time when the chain is terminated. Okay, so that the different DDTPs are going to be fluorescent labeled. Okay, DDTPs are going to be fluorescent labeled. So each time one DD ATP or the C or G or whatever these things are going to be added, different types of colors will be emitted, so that the fluorescent signal will be read by the detector and the detection will be transmitted or processed, and later you can analyze that data using the computer. Okay, so a detector detects the fluorescent signals each time when the chain is terminated. Further, the signals are recorded and analyzed computationally, So the computational software generates various fluorescence peaks depending upon the amount of fluorescence emitted. So again, you might think why this amount of fluorescence emitted is going to be varied. So when the amount of fluorescence emitted increase, the peakness also will go up. Okay. So for example, you see that for the adenine, so there's a color. So there's continuously 3 adenines are in a consecutive manner. Then so A. So the same color is going to be emitted, so that the peak will go up. Okay, 3 times up as compared to a single A. So that then you know that there are 2 or 3 A's or C's like that. So that is the help of the peaks. Okay. So the computational software generates various fluorescence peaks depending upon the amount of fluorescence emitted. So the hypothetical, so now you can see here, okay, so that here the A, and when the peak goes up, so this is for 2C, okay, then a small peak T, and you can see somewhere is there any value for the 3 times. Okay, so here they have shown only for the single and 2 times. So when it goes beyond that level, then you might need to know that is 3 times the same thing is repeated. Okay, so that is the peakness will give you, but the different colors will tell you the different DD trichion, DD nucleotides, and the dideoxy nucleotides. 4 types of nucleotides are there, so that will be having the 4 colors, but the peakness will represent the amount of colors emitted. Then accordingly you will show whether it is a single T or double T. Understood?
Yes, miss.
You can have the page results as well. And then this is for the classical method. Okay, so that as usual, the smallest to the largest one will be reading. So this is for the classical method. Okay, this is for the classical method of the Sanger sequencing. So that again here they say that the principles of the electrophorus stated that the principles you already know. Okay. Okay, then you have a check here. Okay, you have a check here. So when you have the page, okay, the polyacrylamide gel, and you know, if you know that the DNA sequence you are interested in is having 16 bases, okay, 16 bases, then you have to have 16 bands. Okay, so in the gel, if you have 16 bands, then you are pretty sure that the sequencing has worked correctly. Okay, so if you know that the bands are, the spaces are 16, but you are getting only 14 bands, then there is something wrong. Okay, so that you have a check what is the number of sequences of the particular DNA of interest we are looking for, and if you get equal number of bands, then we are pretty sure that our sequencing method has went okay, all right. Clear? So the first point in sequencing the DNA is to match the size of the DNA. If the bands obtained in gel and the nucleotide sequence of our DNA is similar, the reaction is completed properly. For instance, if your sequence is length is around 16, then 16 bands must be present into the gel. So then you can cross check as far as the number of base pairs in a DNA sequence is not. Okay. So the start reading from the smallest to the largest. So that is the Sanger sequencing. Okay, that is Sanger sequencing. So that today I have covered the rest of the transgenic animals and the application of the transgenic animals and the steps involved in the DNA sequencing and the methods of DNA sequencing So under the methods of DNA sequencing, I covered two methods. One is the Maxam-Gilbert method. The other one is DNA sequencing method. Sorry, Sanger sequencing method. So understood, everyone?
Yes, Miss.
Yes.
All of you agree?
Yes, Miss.
Okay, then I will add the video as well because within the lecture I don't have time to play all the videos. I will give you the website address in the LMS. You can just go through it. Okay. Okay.
Yes, Miss.
Yes,
Okay.
Miss.
With that, I conclude today's class. Thank you very much for cooperating with me